Good morning. I would like to welcome everyone to today's MD Anderson Cancer Series in Cancer Center Enjoy Science Summer Seminar. This is a series of lectures given by MD Anderson faculty members and organized by the Office of the Chief Scientific Officer, Dr. Giulio Draetta. I'm Alessandra Ferraioli and I have the pleasure of hosting this series with my colleague, Dr. George Kalin. The goal of the Enjoy Science initiative is to strengthen the connection between the scientific and medical community at the MD Anderson Cancer Center and our colleagues, our patients, and our friends around the world during this time of limited in-person at the bench and in the clinic activities and interaction. This July 17th seminar is being recorded and will be made available at the Enjoy Science website later this week. The speaker of today is Dr. Nidhi Sani. She is an assistant professor in the departments of epigenetic and molecular carcinogenesis and of bioinformatics and computational biology at the University of Texas MD Anderson Cancer Center, Smithville and Houston campus. She holds teaching appointments at Baylor College of Medicine and at the University of Texas School of Biomedical Science, both in Houston. Dr. Sani completed her undergraduate studies at the University of Delhi in India. Subsequently obtained her PhD in biology from the University of Iowa in 2010. Prior to joining MD Anderson in 2016, Dr. Sani was a research fellow in the Department of Cancer Biology at the Dana-Farber Cancer Center and in the Department of Genetics at Harvard Medical School, both in Boston. Dr. Sani is an expert in the field of cancer systems biology and functional genomics. She is the lead author or senior author of numerous high-impact publications in the area of gene regulation and condition-dependent signal transduction in relation to human diseases and several types of cancers. She is the recipient of numerous awards and honors for her research accomplishment. Today she will be discussing for us leveraging system genetics and network medicine for novel cancer therapeutics. Dr. Sani, welcome.
Hello, can everyone hear me?
Yes, we can.
Okay, thank you very much, Alessandra, for the kind introduction. And I'm really thrilled to be here sharing some of the work that my lab has been doing over the past decade or so. And I'm very privileged to have given an opportunity to present at this Enjoy Science seminar series. So I really thank both George and Alessandra, as well as Liz and others who helped set this up. Okay, so the key interest, one of the key areas of interest in my lab is to be able to understand what happens to molecular interaction networks when we introduce disease mutations. And by disease mutations in this context, I refer to as single amino acid change or point mutations or missense mutations. And in order to be able to do that, we need to be able to establish a wild type unbiased reference network of molecular interactions. So by molecular interactions, for a large fraction of my talk today, I'm going to refer to protein-protein interactions as well as protein-DNA interactions. So in order to study signaling network perturbations in disease, we first have to obtain a comprehensive interactome landscape. In order to do this, we embarked on this study to study protein-protein interactions in a large-scale manner in an unbiased way. Here, I'm showing to you the data looking at interactions among the 20,000 proteins in the human proteome. This data was extracted from papers curating protein-protein interactions in a large number of databases such as BioGrid, Intact, String, and others. As you can see here, the proteins are ranked based on the number of publications that they were reported in the literature. And of course, each blue dot represents cases where there was a documented protein-protein interaction. So based on our curation effort of looking at documented protein-protein interactions, you can see a really dark blue zone which tells us there's a large number of protein-protein interactions among proteins that are very well studied, such as our favorite interaction, p53-MDM2, for example. However, on this plot, you see a large sparse zone, which is what we define as the white space, where very few interactions have been identified among proteins. And of course, the sparse zone is very important and interesting because it involves interactions among genes that are implicated in Mendelian disorders, as well as genes that have been identified in genome-wide association studies as important disease-linked genes. So there's a constant need to be able to profile molecular protein-protein interactions in an unbiased way to be able to get enough coverage of these important disease proteins in this sparse zone. In order to do this, we employ a number of assays in the lab to systematically profile protein-protein interactions. And here I'm going to give you a snapshot of some of the assays that my lab employs to study protein-protein interactions. One of the most common assays that we utilize is a protein complementation assay, and we have several iterations of the assay in the lab. The one that I'm showing you here is based on reconstitution of a fluorescence reporter, in this case, the yellow fluorescent protein. The way this assay works is that we fuse protein X or Y, as shown here, to a split YFP protein. And if there is an interaction between X and Y, it will result in reconstitution of the yellow fluorescent protein, and then we measure fluorescence as an output for the interaction. We also have iterations of this assay using a large number of different luciferases, whereby we can measure luciferase activity following protein-protein interaction. Another assay which has been very widely used by us and others in the field is the yeast-2 hybrid assay. And the advantage of the yeast-2 hybrid assay is that it can be used to profile protein-protein interactions for thousands and thousands of proteins because it's very convenient and economical to grow yeast in large numbers. In this particular case, the yeast-2 hybrid version that we use most widely in my lab is based on the reconstitution of the GAL4 transcription factor. We do understand that reconstitution assay based on the GAL4 transcription factor does have its challenges, but it is one of the most widely used assay in the field. We also have assays that look at, for example, of reconstitution of the JAK-STAT signaling pathway in mammalian cells, the MAPIT assay, as well as assays that are looking at in vitro pull-downs in reticulocyte lysates, such as the WEL-NAPA. All of these assays can be implemented in a high throughput manner and allow us to measure interactions for hundreds of protein pairs in a very short period of time. So using a combination of these assays, as I showed you earlier, we were able to profile interactions among the 20,000 proteins in the human proteome. And using this approach, as I said earlier, we found in the literature there was a huge dense zone, but using this unbiased approach of profiling protein-protein interactions, we were able to uncover a systematic binary map of about 14,000 protein-protein interactions between 4,300 proteins. So now we have a framework of these wild-type protein-protein interactions. So the key question that we are interested in asking now is, what happens to this interactome network of wild-type interactions when we introduce point mutations or missense mutations? This is very pertinent to the field of cancer as well as other Mendelian diseases. As you can imagine, with the advent of next-generation sequencing, there are large numbers of genomic variants that are available to us to study. So the first key question we want to understand is, how do we identify which variants are causal variants? And furthermore, how do we functionalize and contextualize these variants in ways where we can look at large numbers of them to prioritize which variants are potential driver variants? And we have quite a few body of work looking at identifying causal variants and furthermore being able to characterize and functionalize these variants in a large-scale manner. And I'm going to describe to you two stories today where we have used a suite of assays to be able to functionalize these variants in a large-scale manner. So the first project that I'm going to describe to you today covers a wide variety of diseases, which we like to call the 1,000 Disease Project. And this covers a large number of missense mutations, paneling a large number of distinct Mendelian disorders. And we got information for these missense mutations from the Human Gene Mutation Database. The Human Gene Mutation Database essentially catalogs germline mutations associated with a large number of Mendelian disorders. So as part of this effort, we generated close to about 3,000 mutations in about 1,200 genes. The way we use this information to be able to characterize these mutations is by using the gateway technology. And the gateway technology is a very powerful way to be able to look at thousands of mutations in many different assays, as I will describe to you. So as part of this study, we did a variety of assays to be able to measure the impact of these mutations in these genes. And the first question that we were interested in asking was, to what extent do these mutations impact protein structure and stability? We know from a large body of work from my mentor, late Sue Lindquist, that molecular chaperones such as HSP90 and 70 are known to buffer genetic variation. Furthermore, we also understand that misfolded and unstable mutant proteins can be salvaged by the chaperone machinery in order to allow them to fold normally to carry out their biological function. Therefore, as part of this assay, we use chaperone binding as an indicator of protein instability or misfolding. In order to measure the extent to which missense disease mutations impact protein structure and stability, we employed a high throughput Lumiere assay. The way this assay works is that we express our wild type or mutant proteins, which we refer to as a BAIT, as flag tagged library of BAIT proteins, which are tagged with the 3X flag tag. We then introduce our BAIT into a luciferase tagged chaperone protein. And in this particular case, we were measuring interactions of our BAIT protein with one of 7 different chaperones. And these chaperones include both cytosolic chaperones such as HSP90 and 70, which I'm going to show you data for in the next few slides, as well as a panel of ER chaperones. If there is an interaction between your BAIT, which is either your wild type or mutant protein of interest, and the prey, which is luciferase tag, we were able to measure luciferase activity, since if there's an interaction, these complexes will be dragged to a bottom of an anti-flag coated 384 well plate. And as a second step, we used a polyclonal anti-flag antibody to quantitate the BAIT concentration. This assay was developed closely with Mikko Taiple and late Sue Lindquist at MIT. So in order to measure chaperone binding, we utilized this Lumiere assay. As here, I'm showing an example of the plot of binding of these BAIT proteins to one chaperone, HSC70, and I'm plotting the wild type interaction score along the x-axis and the mutant interaction score along the y-axis. We, of course, included very well-known clients of HSC70, such as the VSARC protein, as well as a mutation in EGFR that are shown, that are known positive controls and known clients of HSC70. And then we plot our wild type and mutant interaction scores, respectively. Most of the proteins, as you can see on this plot, did not alter their interactions to HSC70 as they lie along the diagonal. However, there were a small fraction of our mutant proteins that do reproducibly increase interaction or binding with HSC70. Furthermore, we found similar profiles of binding to HSP90, where we found that most of these proteins did not show alterations in binding to HSP90 and lay along the diagonal, whereas only a small fraction of these mutations impacted binding to HSP90. So taken together, we found that only a third of the mutant proteins significantly increased binding to one of several chaperones. I'm showing you data here today only for 2, but we measured systematically data for binding to 7 different chaperones. However, a large fraction of these mutations did not radically impact protein stability as measured by binding to these molecular chaperones. So what is the impact of these mutations on the network? So in order to answer that question, we used a combination of 2 protein-protein interaction assays, and I'm showing you here. One, which is a PCA assay. In this particular case, we used a PCA that was based on reconstitution of a luciferase reporter, as well as the yeast-2 hybrid assay, which I referred to you earlier as based on readout of reconstitution of a Galco transcription factor. So using these 2 assays, we were able to define a matrix of PPIs for our alleles, as well as our wild type protein. So as shown in this matrix here, a green box indicates that an interaction has been detected, versus yellow where we lose an interaction. So we measure the interaction, of course, for the wild type protein as well as its associated mutations, and we can expect one of several different outcomes. There will be alleles in which case we do not see any alteration in protein-protein interaction as shown by allele 1. There are alleles such as alleles 2 and 3 shown in this matrix, which lose only a subset of interactions that are measured in these two assays, as well as there are alleles that are more disruptive, such as the allele 4 shown here, that loses all measurable protein-protein interactions as measured by these two assays. So we refer to allele 1 as quasi-wild type allele, alleles 2 and 3 as egetic, meaning that these alleles are losing only a subset of measured protein-protein interactions, and allele 4 being more disruptive are actually alleles that are more like quasi-null behavior that are losing all measured protein-protein interactions. So we wanted to see whether this approach of measuring systematically protein-protein interaction perturbations can help us actually distinguish disease risk variants. So in order to answer this question, we had our matrix of disease mutations from the Human Gene Mutation Database, as I'm showing you here. And in this pie chart, the purple represents the ones that are quasi-wild type that don't change any protein-protein interaction that was measured. The blue are the egetic events, which are losing a subset of protein-protein interactions. And the red are ones that are more destabilizing. And in fact, reassuringly enough, these red part of the pie were also the ones that were in fact binding more to molecular chaperones such as HSP90 and 70, which gave us confidence that these are in fact more destabilizing mutations and in fact were being recruited to bind by chaperones in order to function properly. On the other side of the pie, we looked at non-disease variants that are coming off projects such as the 1,000 genomes, and we saw a very different distribution in the pie as what is shown here. We found that the most number of mutations actually showed a quasi-wild type behavior, whereas we observed perturbation for a very small fraction of the missense variants that came out of the 1,000 genomes. So clearly this gave us confidence that our edgetypes or our edgetyping approach can really help us distinguish disease risk variants from non-disease variants from several databases, 1,000 genomes being one of them, whereas there are other recent studies from the Broad Institute with the NOMAD and EXAC consortium, where our approach can readily be used to able to prioritize risk variants. However, as I'm showing you in the previous slide, we are only measuring protein-protein interactions for a subset, using only a subset of assays. We are not using all possible suite of protein-protein interactions. So we do have a caveat that we may be missing some interactions. However, we do see a large fraction of mutations showing this quasi-wild type behavior. So we wanted to see if there were alterations of other types of molecular interactions, such as protein-DNA interactions. And in order to study that, we used a set of developmental enhancers as our DNA baits. And in this case, these enhancers were derived from the VISTA database that cataloged close to about 500 human enhancers that were studied in embryonic day 11.5 mice. And we measured interactions in a large number of transcription factors to these developmental enhancers. And the approach we used to measure TF-DNA interaction in this case was the East-1 hybrid approach. And very briefly, the East-1 hybrid approach works in the following way. In the East-1 hybrid approach, the DNA bait is placed upstream of a history reporter, and each DNA bait reporter is integrated into the yeast genome. The TF, on the other hand, is fused to the activation domain of GAL4 and is introduced into this DNA bait yeast strain. When the protein binds to promoter, the GAL4-AD moiety activates reporter gene expression, which we measure by growth on selected media. So using a combination of East-1 hybrid assay in yeast, as well as luciferase reporter assays in mammalian cells, we were able to uncover interactions between TF mutations and their associated DNA targets using a combination of these two approaches. And what we uncovered here, as I'm showing you, by our integrated approach of integrating protein-DNA interaction data by these two assays, as well as the protein-protein interaction data that I showed to you earlier in some of the earlier slides, we were able to show that in a large fraction of these mutations, as you can see here, where we in about 35% of the cases, where we did not see any effect on measurable protein-protein interaction as shown by edges connecting these black dots, which are protein-protein interactions, we did observe a reproducible effect as shown by abrogating of interactions with the DNA targets that are in green here. So this really told us that even though in a small space of mutations that we tested, that there is a great benefit of integrating many different molecular interactions to be able to uncover the complete suite of changes caused by missense disease mutations. So as I mentioned to you earlier, the first part of the work was largely focused on Mendelian disease mutations. However, when I first moved here to MD Anderson Cancer Center, we, of course, were in the midst of the tsunami of cancer genomic information coming out at our way through a large number of large-scale sequencing projects, such as the TCGA, as well as the pan-cancer analysis of whole genomes that were being released. So we started this project in here where we wanted to see what is the impact of cancer mutations on on networks. And in order to do this, we started as a subset of genes that are frequent oncogenes or tumor suppressors. And the first question that we were really interested in looking at was looking at mutations in genes that are very well studied, as shown in the graph here, such as EGFR, PIK3CA, BRAF, ALK, PTEN, and many others that we studied as part of this effort. And we wanted to go and see what is the state of art in terms of understanding the impact of cancer mutations in these well-documented tumor suppressors and oncogenes. And we used a combination of three approaches in order to address this question. We first took advantage of the OncoKB data resource that was from the Sloan Kettering Institute, where they went in and curated the impact of disease cancer mutations in many of these known oncogenes and tumor suppressors. Furthermore, we also employed the MD Anderson Institute of Personalized Cancer Therapy resource, PCT, where we go in and manually curate with the help of team of curators at the IPCT, looking at the impact of these mutations in these oncogenes and tumor suppressors. And furthermore, we use a PubMed resource of automated text mining to be able to extract this information. And using a combination of these three approaches, we came to the conclusion that, in fact, the effect of most mutations in these known oncogenes and tumor suppressors still remain largely unknown. This was very reassuring to us since many independent data resource curators help us lead to the same conclusion. So this was the starting point for a study where we in fact took a large number of mutations coming out of the Cancer Genome Atlas, as well as data coming out of sequencing cancer patients at the MD Anderson Cancer Center. And we assembled a large cohort of mutations, close to about 1,120 mutations spanning almost very well studied oncogenes and tumor suppressors, 95 different cases in this case of the wild type genes. And we generated a large library of these mutations and the associated wild type clones. And we measured first the growth factor dependent cell viability of these mutations in two different cell lines, as I'm showing you here. One cell line is BAF3, which is an IL-3 dependent murine cell line. And the other one is a very widely used non-tumorigenic breast epithelial cell line, MCF10A. And using this approach, we were able to measure the impact of these mutations, the ability of these mutations to be able to grow in the absence of either IL-3 or EGF and insulin in the case of MCF10A. And we classified our mutations as whether they are activating or non-activating using this approach. Furthermore, in collaboration with Dr. Gordon Mills, we used the RPPA assay to look at the protein expression alterations caused by these mutations in a panel of cell lines. So using this approach, we were able to classify or characterize the impact of these mutations on viability in these two cell lines. And here I'm showing you the example of just mutations that are plotted in these lollipop plots in the case of EGFR. And we are plotting these mutations that span a wide range of domains in the EGFR protein. And in this histogram below, I'm actually showing you the calls that were made by our experimental approach, which we call the consensus call, as well as calls that were made by a wide variety of computational algorithms that one can employ to just simply put in your key mutation of interest and ask a computational algorithm to predict whether this mutation would have an oncogenic effect or whether it would be neutral. And I hope you can appreciate that clearly that our experimental approach was really able to call out the effect of large numbers of these mutations, just in this case, for example, with EGFR, compared to many of the known computational algorithms which were missing or miscalling some of the effect of these mutations. So there is clearly a big advantage of using our experimental phenotyping approach to be able to annotate the effect of these mutations in these known cancer drivers. Furthermore, we were also interested in asking whether these mutations that were in fact functional and likely activating in our case were in fact, how did they lie in the protein structure? And in fact, for some cases where we had a well-known structure, for example, in BRAF and EGFR, As I'm showing you here, the open ovals reflect the study of mutations that we characterized in our system, and the filled ovals are in fact mutations that were annotated by the OncoKB literature resource. And as you can appreciate that, in fact, mutations that were classified as activating by our system were in fact clustering more closely together in the three-dimensional structure, which also was a very interesting piece of evidence that these were in fact interesting functional candidate mutations in these key oncogenes. So this study has a very massive data resource that has been built, what we call the PHASMIC data resource. And this is available to the community at large in order to be able to assess the effect of mutations on function. And in this database, we have included and integrated data not only from our experimental study of measuring the impact of these mutations on cell viability, but we in fact have also collated data from about close to 17 different computational algorithms that essentially predict the impact of missense mutations on many of these mutations associated in these cancer oncogenes or tumor suppressors. And so one can go in and in fact input your mutation of interest and figure out what impact that mutation may have on activity and compare and see the effect. We also have included RNA-seq data, for example, from the Cancer Genome Atlas for some of these mutations whenever they are available. So in the last half an hour or so, I would like to talk to you about another vignette that my lab has been working on with DNA repair. And in this particular case, I'm going to cover a lot of data today that we've worked on in the case of DNA mismatch repair. And as many of you would appreciate, that DNA mismatch repair is constantly charged with repairing errors that are caused by DNA replication, such as incorrect base incorporation and DNA polymerase slippage, as well as damage caused by oxidation as well as alkylation. The DNA mismatch repair process very briefly begins with the binding of a mut-S heterodimer to DNA mismatches. This DNA mut-S heterodimer is formed by either MSH2, MSH6, or MSH2, MSH3 complex. The MSH2, MSH6 complex essentially is charged with repairing single base mismatches, whereas the MSH2, MSH3 complex is charged with repairing 1 to 2 nucleotide insertion deletions as well as larger insertion and deletions. Upon binding, the mut-S complex undergoes a ADP to ATP exchange-driven conformational change and recruits the mut-L heterodimer to the site of damage. And this mut-L heterodimer is formed by either MLH1, PMS2, or MLH1, MLH3. So we looked at data from about 5,930 exomes across 18 cancer types as shown in this histogram to look at the distribution of MSI high versus MSI low cancers across these 18 cancer types. And as you could easily appreciate, that there was clearly a dichotomous distribution in the microsatellite stability status across these cancer types. Some tumor types, such as the endometrial cancer, which I refer to as USET, and COAD, which is colorectal cancer, were enriched for MSI high cancers, versus other cancers such as ovarian as well as liver cancer were enriched for MSI low. So we know that this deficient MMR process or mismatch repair process, or the microsatellite instability hypermutator phenotype, is thought to produce large numbers of immunogenic neoantigens that can be recognized by immune cells, leading to the approval of MSI status as a clinical biomarker for immunotherapy. We can easily appreciate that while patients do benefit from immunotherapy, often have robust, highly durable responses, more than 60% of these patients fail to respond to immune checkpoint therapy. So here, we sought to identify alternative therapeutic vulnerabilities of this deficient mismatch repair process to fully leverage this deficient MMR, MSI hypermutator phenotype to improve patient outcomes. And in order to study this, we generated a panel of isogenic cell lines by deleting components of the mismatch repair process, as I'm showing you here. We deleted components such as MSH2, MSH6, and MSH3. And as you can appreciate this in the gel up here, looking at DNA repair activity in vitro in these cell lines, where we knock out these components of the mismatch repair pathway, we in fact impair significantly the ability of these cells to repair DNA by mutating, by selectively either knock down or knock out some of these components. However, this ability to repair DNA is in fact restored by overexpression or re-expression of these components back, as shown by re-expression of MLH1 in these isogenic cell lines. So in order to generate a transcriptional signature that is representative of this deficient MMR process, we overlapped differential RNA expression data from Cancer Genome Atlas patients of colorectal, endometrial, and gastric cancer, as well as isogenic cell lines that we generated in the previous slide. And then furthermore, in order to generate therapeutics that can selectively target these MSI high cancers such as endometrial, colorectal, and gastric, we then integrated this data with drug response data in cell lines coming from the Cancer Target Response Database, the CTRP version 2. And using this approach, we were able to identify a very interesting compound called pevnodistat, or MLN4924. So of course, so far what I've shown you is that MLN4924 came out of this correlation, but we wanted to see and experimentally test whether we could really leverage MLN4924 as an important therapeutic target in these MSI high cancers. So in order to do this, we first tested using MLN4924, what will be the impact of treating cell lines in both endometrial as well as colorectal cancer with MLN4924 in both MSI high as well as MSS cell lines of these two cancers. And as you can appreciate here, looking at the viability data from these cell lines, you can see that treatment with MLN4924 selectively sensitized the MSI high cell lines, having little to negligible effect on the MSS cell lines. Furthermore, we wanted to see whether there was any effect of treatment of MLN4924 in MSI xenograft models versus MSS xenograft models of colon and endometrial cancer. And as you would appreciate, that treatment with MLN4924 selectively abrogated tumor formation in these MSI xenografts of colon and endometrial cancer when compared to MSS colon and endometrial cancer. So what is MLN4924? So MLN4924 is in fact an inhibitor of the NED8 activating enzyme NA1. What is NEDylation? NEDylation, in fact, is known to control the activity of colon ring-like ubiquitin ligases, which I'm showing you here as CRLs. And many of these CRLs are known to regulate cell cycle proteins. So the first question we wanted to ask was whether treatment with MLN4924 would cause a cell cycle defect in these MSI cell lines when compared to MSS cell lines. And we went in to address that question, and we did find that there was an effect on treatment with MLN4924 in both MSI cells as well as MSS cells. However, there was no difference in the treatment response. We did observe a depletion of cells in the G0, G1 phase of the cell cycle in cells that were treated with MLN4924. However, this effect was not selective to MSI cells when compared to MSS cells. Furthermore, we also went on to see whether there was increased NEDylation that we observed in MSI patients as well as cell lines. We did observe increased NEDylation in both cell lines that were MSI high, such as HCT116 when compared to MSS, as well as in patient data from the Cancer Genome Atlas. So we wanted to see, in addition to canonical roles of NEDylation controlling the activity of colon ring-like ligases, non-canonical NEDylation can also be induced by stressors that can alter protein homeostasis, such as heat shock and proteasomal inhibition. So we thought that the deficient MMR process may alter the protein homeostasis in the cell, thereby resulting in the accumulation of a large number of destabilized or unfolded proteins that in fact would increase the reliance on NEDylation to allow them to clear from cells. So in support of this hypothesis, we treated cells that were deficient in MMR, for example, in this case, MCF10A cells, where we knocked out MSH2, and we treated them with both bortezomib, which is a proteasomal inhibitor, and a ubiquitination inhibitor, which is MLN7243. And we found that both these inhibitors preferentially targeted these deficient MMR cells, indicating the dependence of these deficient MMR cells on NEDylation to able to carry out their function. Furthermore, we also wanted to see the stress caused by accumulation of these misfolded proteins in the proteomes of these MSI high cell lines of colon, endometrial, and other cancers. And as we can see here, that looking at these MSI high cell lines compared to MSS, we saw an increased accumulation of misfolded proteins as shown by a positive change in the delta G in this plot. So what we concluded from this analysis was that there is an absolutely increased or high number of destabilizing mutation in the proteome of these MSI high cell lines / patient samples compared to MSS. Furthermore, we did in silico modeling of mutational signatures that are very widely used in the field, and we found that in fact this deficient MMR process was highly destabilizing compared to the other mutational signatures. Next, we wanted to use thermal stability shift assays to measure in real time what is the protein stability in the case of these MSI cell lines. So very briefly, the way a thermal stability shift assay works is that it quantifies the change in thermal denaturation temperature of a protein under various stressors. We can measure the alteration in the thermal denaturation temperature in response to pH, salt treatment, drugs, and this assay has been very widely used in the drug industry, for example, to look at off-target effects of drugs as well as drug resistance in cancer cell lines. So using the thermal stability shift assay, we found that in fact the mutant that in fact the MSI HCT116 cells are in fact less stable and have a high number of destabilizing mutations as shown in red compared to the MSS cells. Furthermore, we found that these destabilizing mutations are in fact have a greater impact on protein stability as shown by a greater decrease in the area under the curve compared to neutral mutations which had no impact on the AUC. So taking one example, we wanted to see whether, in fact, this mutant protein in this particular case of this one protein, PTS, we wanted to see whether this mutated protein is, in fact, nedylated when compared to the wild type. And in order to do this, we used IP experiments in which we transfected flag-tagged wild type or mutant P87S PTS and performed immunoprecipitation studies with anti-nedate antibodies. And as you can appreciate, we did see nedylation in this mutated P87S protein when compared to the wild type PTS protein. Furthermore, this nedylation band, which was shifted about 8 kD, was largely abrogated in response to treatment with MLN4924 in this P87 mutant. So this in fact told us that these mutated proteins were in fact nedylated, thereby relying on the nedylation pathway in order to be able to carry out their function. We also wanted to see whether this destabilization phenotype also existed in xenograft models, and we measured the stability of the proteome in these colorectal patient-derived xenografts, and we found that these MSI xenograft models in fact showed a higher or more destabilized proteome when compared to the MSS xenografts. And similar trends were also observed in the case of primary endometrial patient samples, where we see similar effects on MSI having a destabilized proteome when compared to MSS. Next, we wanted to ask whether it's in fact this proteome stability that determines the response to this drug MLN4924. And in order to address this question, we in fact introduced destabilization in MSS cell lines by inhibiting HSP90 or 70. So we introduced MSS cell lines and we treated them with HSP90 inhibitor, HSP70 inhibitor, or MG132. And thereby, once they were destabilized, they in fact became more sensitive to MLN4924. Furthermore, as I showed you earlier in my talk, that in fact destabilizing mutations tend to bind more to chaperones in order to be able to fold properly to carry out their biological function. Based on our previous work, we had shown that in fact destabilizing mutations show an increased binding to a panel of chaperones such as HSP88 or HSP70, HSP90, as well as the panel of other chaperones when compared to neutral mutations. And as you can see here, when we look at data for MSI high versus MSS cell lines of colorectal cancer and endometrial cancer as shown here, there's clearly an increased expression of HSP88, in fact, in both cell lines as well as tumors that are MSI high. So we wanted to see whether if we introduced stabilization of these MSI cancer cells by selectively overexpressing HSP88, whether we could in fact increase resistance to MLN4924. And this was in fact what we saw when we stabilized these proteomes of these MSI high cells such as HCT116, FOCO1, as well as MFE296 of endometrial. we found that in fact these cell lines upon overexpression of HSP88 became more resistant to MLN4924. So this clearly gave us reassurance that protein stabilization in fact determines response to MLN4924. Next, we wanted to see whether MLN4924, what impact would it have if we were to combine this with immune checkpoint therapy. And so we found that MLN4924 is inducing an ER or UPR response, resulting in immunogenic cell death. And we found that using Western blot analysis in these MSI high cells in response to treatment with MLN4924, we saw selective upregulation of markers of ER stress as well as the unfolded protein response. Furthermore, we also found that treatment of cells with MLN4924 also caused increased inflammatory signaling. We found increased CTL cytokine secretion in these cells that were treated with MLN4924, as well as increased immune cell migration. Next, we were wanting to look at immunogenic cell death, and in fact, treatment of these cells with MLN4924, as shown here, showed an increase in immunogenic cell death. And we measured immunogenic cell death in this case by using two different assays. One was looking at the expression of surface calreticulin, and the second was measuring the media ATP concentration. And in both these assays, as you can see, using treatment of cells with MLN4924 caused an immunogenic cell death phenotype. So for finally being able to study whether MLN4924 could synergize with PD-1 in our studies, we used two syngeneic mouse models. And we used one model for MSI colorectal cancer in which we deleted MSH2 from the CT26 colorectal cancer cell line. And for endometrial cancer, we generated a murine endometrial DMMR cell line from a knockout mouse model of MSH2 that was driven by a progesterone receptor expression in C57 black 6 background. So using these murine syngeneic mouse models, we wanted to see whether we can synergize, we observe any synergy between MLN4924 and PD-1. And this is in fact what we saw in these syngeneic mouse models. We found that treatment with PD-1 alone had little to no effect, whereas MLN4924 had significant effect on the tumor volume in these mouse models. However, treatment with the combination of MLN4924 and PD-1 significantly reduced the tumor volume in these mouse models. So this suggested that MLN4924 can in fact synergize with PD-1 and help these patients with MSI cancers. So taken together from this part of the talk, what I've shown you today is that our gene signature guided approach identified MLN4924 as a novel therapeutic target for MSI cancer. MSI cancers, in fact, have increased reliance on nedylation to clear misfolded proteins. And blockage of nedylation causes misfolded protein accumulation, increased inflammation, and immunogenic cell death. And furthermore, MLN4924 is potentiated by immunotherapy and may improve outcomes for patients with MSI cancers. So furthermore, I would just like to conclude by showing some of the other projects that we are doing in the lab beyond the two that I described today. The first part of the talk today covered our efforts on looking at the impact of disease mutations, our functional variomics approach to be able to classify or characterize variants in a phenotypic manner on large scale. We are also looking at understanding what are drivers of this very interesting phenomenon in biology called phase separation. And we have very interesting work right now with both cancer mutations as well as cancer fusions, which are largely unstudied so far on phase separation behavior. We also have a program where we are looking at impact of mutations in the DNA damage repair pathway in a large scale manner. And furthermore, we are also doing CRISPR screens to be able to identify synthetic lethal combinations for combination therapy. And in the end, I would like to say that we have a lot of resources that my lab has built over the years that we are very willing and are able to share with the community, both at the level of the wild type reference Orpheum that I described to you earlier. We have the latest and the most complete Orpheum collection in my lab, as well as a large number of different disease allele Orpheums, part of which I was covering in the first part of my talk, but we have also constantly established more collections and are very open to collaborating and generating these for others who may be interested or working with other labs who may be interested in generating some of these resources and doing these large-scale characterization. So with that, I would like to acknowledge a large number of people here, members of my lab, as well as our bioinformatics collaborators at UT Austin. The second part of the story with the mismatch repair story was largely spearheaded by an awesome postdoc, Daniel McGrail, and my collaborator, Xiaoyi Lin in the Department of Systems Biology, as well as our clinical collaborators who I mentioned along the way. And as well as our funding, we have been very lucky to have received really good support over the years, and would be happy to take any questions if you have any. Thank you.
Thank you very much, Nidhi. This was a wonderful talk. And in fact, by chance, it matched wonderfully with the town hall that we have this morning about the research. Now there were presented young early stage investigators with wonderful record of publication. And in fact, you are the best example of how good these young investigators who are doing basic research coordinated with the clinicians are doing in MD Anderson. The presentation was excellent, and let's start with the question. First, did MLN 4924, it's already in clinical trials, and if yes, what are the results of the use of this drug?
So we are starting, we are looking into the possibility for clinical trials. Right now, it's still early days. I have been asked this question everywhere where I've given this talk, which has been several venues so far. It's still early for us. I think there is definitely a lot of potential for this compound, but I would say stay tuned, and I'll have probably more updates in the near future on where things are.
Thank you. So that basically we have to still to focus on your research. Very, very good. We will do it with pleasure. Now, a second question from my side. You was talking about mutations in coding genes. What's happened and how would you study the mutations who are located in between coding genes and non-coding DNA mutations that certainly are accumulating more and more now with whole genome sequencing studies, and the function is far behind of the understanding for the coding genes?
I think, George, you bring up a really excellent point, and I actually just wrote a grant proposal on this, and I didn't cover that part of my lab. So of course we have a suite of assays, as I said, which I showed only a snapshot of with coding mutations, but there's a large interest in the lab looking at, as you very well pointed out, impact of non-coding variants that are coming out. And they are in fact the vast majority, to be honest, in the genome. And there are many different assays that are out there in the field with the multi-parallel reporter assays, the MPRA assays. We are also using a variant of the MPRA, which is StarSeq, to be able to profile the impact of these non-coding variants. It's still early days for us, but we are actively working towards studying impact of these non-coding SNPs. And it's really exciting and interesting. And we are really at the tip of the iceberg there, looking at some of these non-coding variants coming out of these pan-cancer data efforts from the Peacock Consortium with colorectal cancer, for example. So stay tuned. I hope I have another opportunity in the future to talk about data there.
Thank you very much. Another question. Can RNA-protein interaction be measured with your fluorescence technology?
That is a very good question. And actually, I'm pretty excited about looking at protein-RNA interaction perturbations. We haven't tried that using our technology. However, we are interested in seeing whether we can employ CLIP and other assays, for example, in large scale. We haven't, we've been focused on our networks of protein-protein interactions, but are, of course, interested in either collaborating or exploring ideas of scaling up ability to measure protein-RNA interactions, but haven't done much work there.
Thank you. And one more question. So how did you see in the next future the study of coding mutations? Because there are a lot of mutations who are described, let's say, in 1% of cases, 2% of cases. How would you envision the identification of truly driver mutations, which are also targetable by different therapeutics?
So as I mentioned earlier, there are two sides of the story. I'm sure a lot of people are very interested in going about our experimental approach of really being able to characterize the phenotypic outcomes of some of these mutations. The relatively shortcut way of doing this, as I showed you earlier, is just going into one of these algorithms, and there are several suite of them, like about close to 17 or more, that can actually, if you just go in really quickly and let's say put in the example of your favorite mutation in either TP53 or EGFR, and it can really spit out whether it's really going to have any effect or whether it's going to be benign very quickly. However, we have noticed over the years there has been a constant issue with these. I mean, they are really valuable resources to get quick information, don't get me wrong, but in fact, the problem with some of these computational algorithms is that they are trained around sets of proteins that are very widely studied. They are, again, trained around protein features such as conservation and others that are really biased in some ways. So I think, the short answer is that I think our approach is still very important, and I think it should be employed to be able to characterize any of these variants, whether they are low frequency or high frequency, regardless. I think there's a lot of value in still being able to doing this. I know that there are pooled approaches that have been used by the Broad Institute, for example, in mice, where you can do pooled shRNA screens in mice to pull out potential genes that could be drivers and then go after mutations in them. But I think going with our approach of testing this one at a time variant in a really sensitive manner is the way to go. And I think this needs to be employed more universally to be able to look at variants of unknown significance, for example.
Thank you very much. In fact, this work that you are doing is extremely important because it's an archaeological work, let's say, in this way, that will discover how important are rare, so-called rare somatic mutation. And of course, a lot of people are looking for non-coding mutations. With this said, let's thank you very much, Nidhi. It was a wonderful presentation, very important for all of us scientists, clinician scientists. And of course, I want to thank very much to attendees. Again, I cannot say more that how important you are, because without you, and you are in hundreds present to webinars all over the world and in MD Anderson, without you, these webinars cannot take place. I want to thank also the people who organize and help us, Liz Kent and Ellison Garth, and of course, let's see you guys next Friday. Friday is the day of Enjoy Science. Till then, in fact, enjoy science, make discoveries, and keep in mind, science is a way to reunite people. Doesn't matter the color of the skin, doesn't matter the country of origin, because we are all here in this wonderful institution and in this wonderful country where only the sky is the limit for science, to make discovery, to help the patients, and basically to move forward the medicine. Today you listen a wonderful talk who it's quintessential proving all of this. Thank you again and good luck to all of you, and see you next week.